the microbe can infect the cell. Causes the cell to bring it in through endocytosis. But I mentioned that we have this sampling system that we use to check bacteria that are in the environment regularly. In this process called transcytosis. And basically what happens is that the cells, we have certain cells that will sample bacteria that are in this lumen here. And so it samples that bacteria and it delivers it to the basal side of the membrane. And over here, we have, our macrophages moving along doing their thing. And so this microbe gets delivered to the other side of the mucous membrane. And the macrophage, of course, reaches out and engulfs it. But this particular microbe has the ability to avoid digestion in the phagosome. And what it'll do is it'll break free from the macrophage. It'll infect other cells from the backside now. And from here, move into neighboring cells as the infection spreads. Taking advantage of our transcytosis sampling process, where normally we would sample bacteria, the macrophage would engulf it and break it down or present antigens to T cells or something like that. But in this case, this particular microbe is able to avoid digestion in the macrophage, spread to other cells, and continue the infection. Salmonella employs this technique. Avoiding host defenses. One of the things that you can do is you can hide inside the cell. And that's exactly what's going on right here. If you hide inside the cell, you don't have to worry about the macrophages coming along and destroying you. You don't have to worry about being tagged by antibodies and complement. So how do you get identified and destroyed? I mean, how are we going to address this type of infection if it's only inside the cells?
Kind of like the virus itself.
I'm talking, there you go. Yeah, our cytotoxic T cells, and maybe even our natural killer cells, are going to come along, and what's going to happen, right, is that these cells that are infected, their surface receptors are going to change. They're going to be presenting then products from the bacteria on the surface to cytotoxic T cells likely, that'll come along and destroy these cells. So they can hide in cells for a while, but they're going to mark those cells that they're hiding in. And so our cytotoxic T cells can come along and will eventually destroy those cells. Otherwise, we'd never get rid of that kind of infection, would we?
So would that be one of the toll-like receptor things then?
Like
Right there, toll-like receptors, would they be putting something out on the surface?
In this case, they're not proteins that are inside presenting bacterial products to the surface. Remember, they're like toll-like receptors, but when they're presenting internal molecules, we call them nod proteins. Okay. NOD. It's the same thing as a toll-like receptor. The difference is toll-like receptors are external.
And nods on the inside.
Exactly. So you can hide in the cell, avoid complement. And We've talked about this already. You can avoid complement by shedding your pili, changing the antigens that you might have on the surface. Avoid destruction by phagocytes. There are a number of ways that you can do this. A capsule itself can sometimes provide you some protection. Makes it difficult to be recognized. Several microbes have proteins on the surface that can interfere with antibodies so that they can't bind. We've mentioned proteases. A number of microbes produce proteases that can prevent complement and antibody binding to those cells. And a lot of phagocytosis requires that those cells have been marked by antibodies. We've mentioned already here with this process of transcytosis, if you can avoid being digested inside the macrophage, so if you can avoid the phagosome fusing with the lysosomes. These are ways that you can avoid destruction by the phagocyte. Avoid antibodies. Again, same thing that we've discussed already. Shed your pili, antigenic variation, production of proteases. Questions?
You said shed pili, antigenic variation, and
Proteases? So, a disease has to do damage to the host. So, the damage that's done can be limited, like we've talked, can be local or can be systemic. Generally, though, when damage is being done, it's facilitating the dispersal of the microbe. You think about that. I mean, the cells that are being destroyed, a lot of times are being destroyed because the microbe is causing the destruction in order to be dispersed. Much of the damage that is done by microbes is done by exotoxins. They may act locally, they may be carried throughout the body. Our body produces antibodies to these different toxins. And there's several types. I'm going to mention 3 of them here. An AD toxin is a toxin that has 2 parts. One part identifies the cell for infection, and the other part causes the infection, or causes the damage, if you will. So the B part in AD toxins identify the cell, adhere to the cell, and then the A part gets delivered either because the B causes the cell to bring it in, or generally that's the way it's going to happen. Sometimes the whole bacterium is brought in. The A part of the toxin then causes the internal change that takes place, causes the damage that takes place to the cell. Membrane damaging toxins, names pretty self-explanatory. They disrupt the plasma membrane of the cell. And a third type, superantigens. Now, a superantigen is a microbe that causes a T cell response in excess of what we would want. So let's imagine that normally 1 in 20 T cells might be active for any particular infection. But if that infection turns on more, say it turns on 5 of those 20 cells, that overexcitation of T cells, if you will, can cause an increased production of cytokines and moderators and chemicals that damage our cells. See, the chemicals that our immune system releases doesn't always just target bacteria. They'll also target our own cells. And so in the case of a superantigen, what you have is a toxin that is actually linking a couple of T cells together, turning them both on. And so now you've got a lot more cytokine production, and it turns out to damage the cells that are being exposed to that production. Last type of toxin I'll mention here, and this is one we've already talked about, endotoxins. These then are not being secreted, but are actually part of the microbe. They're part of the gram-negative cell wall. And they too can cause an immune response. A vigorous immune response, depending on the number of bacteria that you get exposed to. And I'm so frustrated with our computer. So I'm going to assign you guys to go to YouTube and watch a video on acute inflammation. Watch a video titled The Process of Phagocytosis. No, you can write it down just as quickly as I can. A video titled Immune System, Natural Killer Cells. One titled Flu Attack. And one titled T Cell Activation. They're all short 2-3 minute videos, won't take you long to watch them. And feel free to expand and look at some of the others that are there too. B cells, T cells, natural killer cells, macrophages. Lots of different videos there for you to look at. I have acute inflammation, process of phagocytosis, immune system, natural killer cells, flu attack, T cell activation.